Good morning, class. Okay, before we formally start our discussion, may I call on our class president to lead our prayer? Okay, so who's absent today? Okay, before we proceed to our topic, let me ask you about what you already know in genetics. Anyone? Okay, very good. What else? Correct. So, have to show you pictures and tell me what you see. Okay, tell me what you see in these pictures. Very good. Okay, so genetics. Genetics is a scientific study of genes and heredity, of how certain qualities of traits are passed from parents to offspring as a result of changes in DNA sequence. A gene is a segment of DNA that contains instructions for building one or more molecules that helps the body works. Okay, everyone, who can define inheritance? Okay, so how can we trace the inherited genes? Very good. So, Gregor Mendel. Who knows Gregor Mendel? Huh? Anyone? So, Gregor Mendel is an Austrian monk, the father of genetics. So, Mendel was an Austrian monk and a father of genetics. He worked with a different true breeding or pure breed pea plant. So, as I told you to advance read our topic, class, why did Mendel use pea plant to study genetics? Anyone? Yes, very good. Because pea plant were good at self-pollinating and seed in one season. And the last one is have many different true breeding type. Okay. So let's proceed to a Punnett square. Punnett square is a square diagram that used to predict the genotypes of particular cross or breeding experiment. Here, I'll show you an example of this using this Punnett square. So, this one. So, our example is a heterozygous smooth pea pod plant is crossed with a wrinkle pea pod plant. There are two alleles for pea pod smooth and wrinkle. Protect the offspring from this cross. Here is the example. This one. Okay. This is the heterozygous smooth pea pod and a heterozygous wrinkled pea pod plants. The result is two heterozygous and two homozygous. And 75% of this result is smooth pea pod plant. Okay. Okay, now let's proceed to a non-Mendelian inheritance. Non-Mendelian inheritance, we have incomplete dominance, multiple alleles, codominance, polygenic traits, and environmental traits. Okay, first, incomplete dominance. Incomplete dominance is the no dominant allele is expressed. And multiple alleles, more than two alleles can express. And a codominance is two dominant alleles can be expressed for the same trait. Okay? Okay, the next is polygenic traits. And polygenic traits, several genes or genotypes can influence in a particular traits or phenotypes. Okay. And the last one is the environmental factors. And environmental factors, the phenotypes or the phenotypes can be influenced by the environment. Okay, class. So, the difference between Mendelian and non-Mendelian genetics is that the Mendelian traits are determined by dominant and recessive alleles of one genes. On the other hand, non-Mendelian traits are not determined. The Mendelian genetics or Mendelian inheritance is determined by the alleles determined by dominant and recessive alleles, while the non-Mendelian is not determined by dominant and recessive alleles. In non-Mendelian inheritance, it can be governed by more than one genes. So class, okay, what do you think are the similarities of Mendelian and non-Mendelian inheritance? Okay, very good. Are there any questions about this? Okay, class, so let's proceed to our next topic. We have advanced genetics. So, chromosomes. Chromosomes is a DNA molecule is packaged into the thread-like structures made of proteins. So, types of chromosomes. In type of chromosomes, we have here a metacentric, submetacentric, and acrocentric, and telocentric. So, chromosomal abnormalities. So, in chromosomal abnormalities, we have numerical or structural abnormalities. Okay, this is the chromosomes that are missing, switch to another chromosomes, extra, or torn upside down. Okay. So, structural abnormalities. Okay, class. So, in structural abnormalities, it has deletion, duplication, insertion, and ringing. And numerical abnormalities naman is we have monosomy. Okay. So, Okay, do you have any question about this? Yes? Okay, simply because numerical abnormalities are whole chromosomes, either missing or extra to the normal pair. While the structural abnormalities are when part of an chromosomes is missing extra, switched to another chromosomes, or turned upside down. Understood? Okay. So our next topic is the population of genetics. So what does the population of genetics means? Anyone? Any idea? Okay. So population of genetics, it is a distribution and change in frequency of alleles within populations. Population of genetics includes here the the adaptation, speciation, and population structure. Okay. So, based on several processes of evolution, we have here the natural selection, genetic drift, gen flow, and mutation. Okay? Okay, class. So, is there any question regarding population of genetics? If you say so, then let's proceed to the next topic. We have here a molecular genetics. So, nucleic acid. It is the rule of nucleic acid. So, nucleic acid is a naturally occurring chemical compound that serves as the primary information. Nucleic acid is carrying molecules in cell, and they play important role in directing protein synthesis. Okay, class, did you know the two main classes of nucleic acid? Yes? Very good, correct. Okay, so I have here the three major components of DNA. So, class, I know that you already knew about this three major components of DNA. Are you? Okay. Okay, then. So, I have to ask you some questions. So, what what did you learn from today's discussion? Yes. Okay, then why is it important to study genetics? Okay, class. So now we have a activities. So, so we have a role playing. So, create a 3 minutes role play that shows the important importance of studying inherited genetic characteristics or traits from the parents down to their offspring. So here is the rubrics. Our rubrics is the content is 50%. The delivery is 20%. The acting skills is 30% for the total of 100%. Okay? And now, class, we will having a assessment, which is you have to answer the following questions. Okay, write your answer in a 1/2 crosswise yellow paper and submit it to me if you are done. Okay, and for your assignment, for your assignment is okay. Your assignment is create a 500 words essay containing of what have you learned in genetics. And this is our rubrics. The content is 60 points, organization is 30 points, and the grammar is 10 points for a total of 100%. 100 points. Okay, and that's all for today. And I hope you learn a lot about our discussion. Bye.